I want you to completely erase what you think you know about aging. Just wipe the slate clean.
Yeah, totally clear it out.
Right. Forget the wrinkles, forget the stiff joints, and, forget cognitive decline for a second. What if the exact same cellular mechanism that we actively blame for tissue degeneration as we get older is, well, the very mechanism that secretly built the security systems of your brain before you were even born?
It sounds like science fiction, honestly.
It really does.
Yeah.
Usually when we talk about biological aging, there's this expectation of decay. It's framed as a failure of the system. But when we look at the very foundation of human development, the lines between decay and creation blur in a way that is frankly mind-bending.
They do. And that's why today we're doing a deep dive into a really groundbreaking 2026 article from the journal Cell. It's by Watson and colleagues.
Right. And our mission for this deep dive is to investigate every single conceivable detail of how these senescent or aging cells actually act as programmed embryonic architects.
Which is wild.
It is. To build the blood-brain barrier and the blood-cerebrospinal fluid barrier. Okay, let's unpack this, because we are diving into a massive paradigm shift in biology here.
It truly is a profound paradox. I mean, we are taking a concept inherently associated with cellular exhaustion and repositioning it as this engine of early life creation.
Yeah.
And to really understand human health, and especially how we treat diseases later in life, we have to connect these microscopic embryonic discoveries to the bigger picture.
So let's start by defining what we are actually looking at, because when you hear the phrase cellular senescence, the classic textbook definition paints a pretty grim picture.
Oh, very grim. A zombie cell.
Exactly. It's usually described as a stress response. The cell gets damaged, its DNA frays, so it slams on the brakes to avoid mutating into cancer. It doesn't divide, it doesn't die, it just sits there secreting inflammatory garbage locally.
Right, but what this paper is arguing is that in the developing embryo, that definition completely falls apart. It falls apart because the context is entirely different.
Yeah.
You really have to look at the genetic drivers. So in an aging adult, senescence is typically driven by a stress response protein.
That's P16, right? Or CDKN2A.
Exactly, P16. It's a reaction to accumulated damage over a lifetime. But embryonic senescence, the kind that's actively building your brain, is highly regulated. It's programmed. It is specifically driven by a completely different protein called P21.
Also known as CDKN1A.
Yes, P21. And it operates totally independently of that P16 stress pathway.
Which is a huge distinction.
Massive. This P21 dependency is the molecular signature telling us that, hey, this isn't a damaged cell clinging to life. It's a tool being deliberately deployed by the embryo.
Okay, I want to make sure I'm visualizing this correctly. We have these P21-driven cells, but how do scientists actually prove a cell is truly senescent and not just, temporarily inactive? Reading through the paper, there seem to be 4 distinct levers a cell has to pull.
Right, the hallmark.
Yeah, to be officially recognized as a senescent architect. And I actually made a mnemonic for you listening to help remember this. Think of CTLS cells that linger secretly.
Oh, I like that. CTLS. Let's break those down.
So the first lever, the C, is cutting the engine, cell cycle arrest or exit. It permanently stops dividing.
That's the foundational step. But cutting the engine isn't enough on its own. The second lever, the T, is the transcriptional signature, specifically the SATS, or senescence-associated transcriptional signature.
So they're basically rewriting their software.
Exactly. If we look at the RNA being produced by the cell, it matches a highly specific pattern. It stops expressing genes for cell division and radically upregulates genes for tissue remodeling.
Okay, so the engine is cut, the software is rewritten. The third lever, the L, is where it gets highly microscopic. Lysosomal dysfunction, stockpiling specific trash. Now, lysosomes are the cell's internal recycling plants, right?
They are. And in a normal, healthy cell, lysosomes operate at a highly acidic pH around 4.5 to break down cellular waste. But in a senescent cell, these recycling plants become engorged. They're altered. Their internal pH rises slightly to about 6.0.
So they become less acidic.
Right. And because of this specific alteration, they accumulate massive amounts of a specific enzyme called SA-beta-galactosidase.
SA-beta-gal.
Exactly, which notably remains active at that higher pH of 6.0. They also stockpile lipofuscin.
Which is basically what? Indestructible cellular garbage?
Pretty much. It's oxidized lipid cellular trash. So finding that specific enzyme activity at pH 6.0 is the gold standard biological marker that a cell has entered true senescence.
Which brings us to the fourth and arguably most important lever in our CTLS acronym, the S, the broadcasting tower. or in the paper's terms, the SAP.
Right, the senescence-associated secretory phenotype. Say that 3 times fast.
Yeah, no thanks. But this is where the magic happens, right?
It really is. Senescent cells don't just sit there quietly hoarding trash. They're intensely communicative. They turn on their broadcasting tower and secrete a massive cocktail of cytokines, chemokines, and growth factors
into their immediate environment.
Yes. This secretory phenotype is how they actively, physically remodel the tissue around them. They signal neighboring cells to migrate or change shape or build new structures entirely.
So to be a true architect, you need all 4. Cut the engine, rewrite the software, stockpile the specific trash at pH 6.0, and turn on the broadcasting tower.
All 4.
And the reason we have to be this strict is because of imposter cells.
Oh, the Cachorrezia cells.
Yes. The researchers looked at these special neurons in the developing cortex, and they expressed the P21 protein, and they completely cut their engine. They stopped dividing.
So they checked the first box.
Right. If you just looked at the first lever, you'd say, aha, a senescent cell. But they don't hoard the trash, and they don't broadcast the signals. They just use P21 to temporarily pause their cell cycle so they can mature into a neuron. It's like wearing a hard hat doesn't make you a construction worker.
That is a vital distinction. The genuine P21 positive senescent workers identified in this study are 3 distinct populations. You have the choroid plexus epithelial cells, which we'll call Chupo cells.
Okay.
You have the vascular endothelial cells, or VECs. And you have brain-resident macrophages. These 3 groups actively pull all 4 levers.
Okay, so if we just do a quick 5-minute recap and sub-summary bullet for you listening, senescence isn't just aging. It's an embryonic tool defined by 4 strict hallmarks: cell cycle arrest, transcriptional changes, lysosomal dysfunction, and a secretory phenotype. CTLS. And it makes you think, think about a time you repurposed an old tool for a brand new job. How does that change how you view the so-called aging proteins in your own body?
It really makes you look at your own biology differently. We spend so much time trying to scrub away the signs of aging, but these exact same biological pathways were the highly regulated tools that built our neural infrastructure.
Right. And here's where it gets really interesting. To understand how they actually pull that off, we have to look at the spatiotemporal dynamics, the timeline.
Yes, the timing is everything.
And the way the researchers tracked these cells is fascinating. They used an inducible lineage tracing model. It's called P21 Crier T2.
It's a brilliant piece of genetic engineering, honestly.
It is. So if I'm understanding the mechanism here, they engineered pregnant mice so that if the researchers injected a drug tamoxifen at a highly specific moment in pregnancy,
say embryonic day 11.5 or E11.5.
Right. Any single cell expressing the P21 gene at that exact second would be permanently tagged with a glowing red fluorescent protein.
It's like dumping a bucket of permanent invisible ink that only sticks to the active construction workers and then turning on a black light.
I love that analogy. And they could track those glowing cells all the way to postnatal day 21 or PND 21.
And that tracking revealed 2 completely different construction strategies in the brain.
The temporary crew and the permanent crew.
Exactly. The first strategy utilizes a transient temporary crew. This crew is made up of the vascular endothelial cells, the VECs, which line the blood vessels, and the brain-resident macrophages, which are immune cells.
And together they build the blood-brain barrier.
Right.
But wait, let me stop you there. To build this barrier, the paper notes that they utilize a pro-inflammatory broadcasting signal. Their SAS is inflammatory. We're talking about active inflammation inside a developing embryo. That sounds incredibly dangerous. Wouldn't that damage the fragile tissue?
I know. It sounds totally counterintuitive. We are so conditioned to view inflammation as collateral damage or a disease state. But in this developmental window, peaking around embryonic day 14.5 to 15.5, during hindbrain and forebrain angiogenesis, localized inflammation isn't destructive.
It's not.
No, it's a mechanism of rapid targeted change. It's highly cooperative. The senescent endothelial cells act as the signal callers. They secrete specific inflammatory factors like CSF1 and IL-34.
Biochemical breadcrumbs.
Exactly. And the macrophages possess the exact receptors for those breadcrumbs. So the senescent blood vessel cells actively recruit the immune cells, and together they coordinate the complex physical branching and weaving of the brain's vascular network.
They lay down the scaffolding.
Yes.
And then what absolutely blows my mind is that by postnatal day 21, the senescence markers in these vascular and immune cells completely vanish.
They're just gone.
They do their job, they build the physical walls of the blood-brain barrier, they turn off their broadcasting towers, and they exit the senescent state. They were literally temporary contractors.
But then you have the permanent crew.
The choroid plexus epithelial cells.
Right, the choroid B cells.
If the temporary contractors build the walls, the choroid P cells are the permanent water filtration plant operators. They form the blood-CSF barrier, which produces the cerebrospinal fluid that physically cushions and chemically nourishes your brain.
And their temporal dynamic is entirely different from the temporary crew. Their senescence starts early, right around E11.5. By day 13.5, about 50% of these choroid plexus cells are tagged as actively senescent.
Yeah.
And the crucial finding, they stay that way.
Wherever.
Well, the researchers found these fully senescent SA beta-Gal positive P21 positive cells functioning in the brains of mice that were 12 months old.
Wow.
That is late adulthood for a mouse.
So a cell enters a paused aging state as an embryo and just stays there for its entire life.
It appears so.
And they don't use that inflammatory toolkit either, right? Their broadcasting signal, their SASP, is totally customized for this permanent job.
Yes, it's a non-inflammatory SAP. They secrete factors that drive ciliogenesis,
which is the growth of those tiny hair-like structures, the cilia, that constantly sweep and move fluid around.
Exactly. They also heavily upregulate ion transport, literally filtering the blood to create pristine cerebrospinal fluid.
It's a profound dichotomy.
It really is. The brain utilizes temporary inflammatory senescence to quickly build the blood vessels, but it relies on permanent non-inflammatory senescence to maintain fluid production.
Okay, so let's hit our 10-minute recap and sub-summary bullet. Brain barrier development uses two strategies: temporary inflammatory senescence for blood vessels and permanent non-inflammatory senescence for CSF production.
Nailed it.
And here's a question for you listening. If you had to design a security system for the brain, would you rely on temporary contractors or a permanent guard?
It's amazing that your body chose both.
It really is.
Yeah.
But of course, correlation isn't causation.
No, it's not.
Seeing glowing red cells near a structure doesn't prove they actually built it. So to prove that these senescent cells are the real architects, the researchers had to break the system.
They had to see what happens when you remove them. The ablation experiment.
Yeah.
This is where the biological mechanics become undeniable. It's a real medical detective story. The researchers asked what happens if we genetically delete these P21 positive workers right in the middle of mid-gestation.
And to do this, they used a model called P21-IDTA. Now, DTA stands for diphtheria toxin A. I have to admit, when I first read that, I was super confused. Diphtheria is a deadly human disease. Why on earth are researchers introducing a severe pathogen into a developing mouse embryo?
Right, it sounds terrifying.
Yeah.
But they aren't introducing the bacteria itself, they just borrowed its weapon.
Okay.
They genetically engineered the mice with a highly specific kill switch. When the researchers administer a triggering drug, any cell that is actively turning on the P21 gene suddenly produces this isolated diphtheria toxin protein internally.
Wow.
Yeah, causing that specific cell to instantly self-destruct. It allows them to surgically wipe out only the senescent cells without touching anything else.
So it's like a molecular sniper rifle.
Exactly.
So they pull the trigger, wipe out the senescent workers, and what happens?
Catastrophic failure. By E18.5, over 75% of the embryos lacking these senescent cells suffered from severe meningeal or cerebral hemorrhage.
Brain bleeds.
Massive ones. Red blood cells were physically leaking outside the vessel walls, or lumina, pooling into the brain tissue. The blood-brain barrier didn't just weaken, it structurally failed.
But the how is the fascinating part. Because initially, I assumed that if you kill the architects, the blood vessels just wouldn't grow at all.
A logical assumption.
Right, but that's not what happened. By day 15.5, the sheer volume of the blood vessels, their coverage, actually increased, but their valency plummeted.
Yes. Valency is basically a mathematical way of describing the complexity of a network, how the branches actually connect to form functional loops and tight junctions.
Think of it like city planning. Without the senescent cells there to control the process, the other endothelial cells proliferated uncontrollably. They just kept multiplying.
Because the P21 cells were the brake pedals.
Exactly. They built miles and miles of extra roads, but they never stopped to build an intersection. Without the P21 workers, the cells never paused their division long enough to mature and properly connect the tissue.
And because they were dividing so frantically, the extracellular matrix The ECM, which is the biological mortar that holds the vascular bricks together, was dumped chaotically.
Just a mess.
A total mess. We see structural components like laminin alpha-4 and collagen IV abnormally elevated. They were overproduced, poorly organized. So the vessels became structurally unsound, leaky tubes. That algorithm of failure explains the fatal brain bleeds.
But that's just the temporary crew. What happened to the permanent crew, the choroid plexus, the water filtration plant?
It suffered a massive physical collapse. The lateral ventricle, which is the large, fluid-filled cavern in the brain where the choroid plexus resides, literally shrinks inward. Ventricular collapse.
Why does it shrink, though? Just because the cells died?
No, it's a functional failure.
Yeah.
It shrinks because of a catastrophic drop in a specific marker called APP1, or aquaporin-1.
Okay, what is that?
Aquaporins are essentially plumbing channels that allow water to pass through cell membranes. The choroid plexus uses them to pull water from the blood and secrete it as cerebrospinal fluid.
Oh, I see.
Without the senescent cells orchestrating this, aquaporin-1 levels plummet. And if you can't pump water into a balloon, the balloon deflates. The ventricle collapses.
And it's a compounding disaster, right? Because the blood vessels are also failing, the marker for blood supply, CD31, drops dramatically inside the choroid plexus.
Yes, a severe loss of CD31 positive area.
So it's losing its structural integrity and its blood supply simultaneously.
And ironically, while that structural protein we mentioned, collagen phi, was overproduced in the brain's main blood vessels.
Yeah.
Inside the choroid plexus, the intensity of collagen phi was severely reduced. It showed impaired ECM integrity. The biological Jenga tower was collapsing from two different directions.
It's a perfect metaphor. So let's do our 15-minute recap and sub-summary bullet. Deleting embryonic senescent cells results in massive structural failures. You get brain bleeds, poor vascular branching, and collapsed ventricles.
Total devastation.
And the question for you listening. Think about that biological Jenga tower. Which missing block do you think is more dangerous? The loss of the physical blood supply branching to the tissue, or the loss of the spinal fluid pressure cushioning the entire brain?
Losing these senescent cells pulls out both blocks at the exact same time.
It's absolutely wild. So what does this all mean? This forces us to confront a massive pivot in how we view human biology. We have to completely abandon the binary view of cellular senescence.
We truly do. I mean, for decades, science has treated senescence as a biological switch with only two settings. It was either a strictly transient, temporary benefit, like preventing a tumor or closing a wound, or it was a chronic, toxic pathology that drives aging and age-related disease.
Right. Good or bad, short-term good, long-term bad.
Exactly. But these CHAPI cells completely shatter that binary. They prove that senescence can be a highly regulated, wildly beneficial, lifelong physiological state.
Especially for post-mitotic secretory cells, cells that need to pump out fluids and proteins for decades without ever dividing or regenerating. They enter senescence to gain a structural superpower, ultimate stability.
And if we connect this to the bigger picture, this discovery sends a massive shockwave through the field of neurodegeneration. Think about how we are currently approaching diseases like Alzheimer's, Parkinson's, and tauopathy.
Yeah, right now, one of the most heavily funded areas in biotechnology is the development of senolytics.
Exactly.
Drugs specifically designed to hunt down and chemically clear out senescent cells from the aging body. It's pitched as the anti-aging holy grail.
Because the logic has always seemed sound. You know, if aging cells hoard toxic trash and broadcast chronic inflammation that causes brain fog and tissue decay, let's just flush them out. Erase the zombie cells.
But this paper provides a massive, glaring warning sign. If we use systemic senolytics to blindly clear out all senescent cells in the brain, what else are we hitting?
We might inadvertently destroy the choroid plexus. We could chemically obliterate the permanent blood-CSF barrier that has been keeping the brain mechanically cushioned and chemically balanced since we were fetuses.
You'd be blowing up the city's permanent water filtration plant in a blind attempt to clean up some trash in the streets.
That is exactly what you'd be doing. The collateral damage could be far worse than the disease itself. Medical interventions targeting aging cells will now require an unprecedented level of molecular precision.
We can no longer afford to just look for the SA-beta-gal enzyme and attack.
No, we have to distinguish between the maladaptive inflammatory senescent cells that accumulate in Alzheimer's plaques and the protective non-inflammatory senescent cells that act as permanent load-bearing pillars in the brain.
Which brings us to our final recap bullet. The concept of cellular senescence must be modular, not binary. Targeting aging cells medically requires extreme precision to avoid destroying permanent barrier cells.
It changes the entire paradigm of how we view the timeline of the human body.
And my final question for you listening: how do we fight the diseases of aging if the exact same biological software is actively keeping our brain fluid clean right now?
It's a tough question.
It is. Today, we've journeyed from the, wow, you break your arm, the x-ray is clear. Broken or not broken.
Right.
But the software of your own survival, the very mechanisms keeping you alive today might be the same ones aging you tomorrow. That is beautifully, dangerously murky. Keep diving deep, and we'll see you next time.